Hi everyone, welcome to the second lecture of the urinary system. Today we're going to start looking at the processes that we learned the names of in the previous lecture. We're going to start looking now at how some of those things actually work. So let's go ahead and start, as we always do, with our not really attendance questions. First, what happens at the arteriolar end of a capillary? Next, what happens at the venous end of a capillary? What is found at each end of a glomerular capillary? And what is the process that removes substances from the blood inside of Bowman's capsule? Go ahead and try to answer those. Pause your video. Hopefully you've done that, so let's go ahead and see if we can find the answer to these questions. Now, these first two questions go all the way back to when we first started talking about the cardiovascular system, when we were looking at capillary fluid exchange. So what happens at the arteriolar end of a capillary? Well, this is when substances were moving out of a capillary into the interstitial fluid of a tissue. And at the venous end of a capillary, this is when waste was moving into the blood, into the capillary from the interstitial fluid of a tissue. Now, what is found at each end of a glomerular capillary? Well, remember, a glomerular capillary, that's the special type of capillary that's found inside the kidneys, also just called a glomerulus. These are very special capillaries that have an arteriole at each end, and that's going to come into play today. What is the process that removes substances from the blood inside of Bowman's capsule? That is filtration. And filtration is going to be the topic of today's lecture. So all of these things are going to really be important in today's lecture. If any of this is not familiar, go back, make sure that you familiarize yourself with them before continuing with today's lecture. So filtration. Filtration is a passive process. It's going to be based on pressure, and it's going to be driven by things like hydrostatic pressure, osmotic pressure. It's going to be pushing things out of the blood into Bowman's capsule. What is or is not filtered? Things that are smaller than 3 nanometers are filtered. Those are things that are incredibly small. Things that are between 3 and 5 nanometers, some of them are filtered, some of them are not, since they are a little bit bigger. Now that's still very, very small, but they are in that zone that some of them are filtered, some of them are not. Things that are 5 millimeters and bigger, those things are not filtered, they're too big. And anions. Anions are not filtered because of their charge. Anions are negatively charged, so they do not pass through. Because of their negative charge, they are repelled. So what we see over here is our glomerulus inside of Bowman's capsule. Remember, glomerulus plus Bowman's capsule, this is called a renal corpuscle. Here we have the afferent arteriole bringing blood in. That blood passes through the glomerulus. As it is filtered, the blood, then some of the liquid portion is then sprayed out into Bowman's capsule. Anything that is not filtered then leaves through the efferent arteriole. Here we have a very magnified image of that glomerulus. The red thing is the actual capillary. The yellow thing are special cells called podocytes. The little black dots, those are holes in the capillary walls called fenestration slits. Those are actual holes in the capillary that the liquid is going to spray through. I always say imagine a garden hose that someone has poked holes in. So as the blood goes through the capillary, just like water going through that garden hose with holes in it, that liquid is going to spray through. Well, these podocytes have these extensions called foot processes. And those foot processes reach across the surface of that capillary, and they move those foot processes over those fenestration slits. And they will either cover or uncover those holes as needed, letting more or less of that fluid out as needed. So most of this lecture, what we're going to be doing is drawing how filtration works. And what we're going to be drawing should look very, very familiar. And as a matter of fact, what we're going to first start with is a drawing that we have seen before. This is a drawing that we looked at way back in capillaries. This is capillary fluid exchange. When we drew what happens at a capillary, this is the final image that we saw there. This is not what we're drawing now. This is what we ended up with when we drew capillary fluid exchange. This was the arteriolar end of a capillary, the venular end of a capillary. As blood flowed through that capillary, at the arteriolar end, fluid was moving out. At the venous end, fluid was moving in. We had an outward pressure of 10 mmHg. At the venous end, we had an inward pressure of 8 mmHg. So that meant that fluid moved out, but not all of it moved back in. So we were supplying down here. We were reclaiming down here, but not all of the liquid that moved out was reclaimed. We said that that fluid that was not reclaimed was returned by way of the lymphatic system. So that was how a capillary worked. Now, since we are dealing with a capillary when we look at the glomerulus, all of this is going to come into play again. So let's take this image. If you have this drawn, if you have this handy, go ahead and grab this image. Set it aside because we're going to draw something that looks very, very similar and you can reference it. This is the image that we're going to start with, and let me describe the similarities and the differences. Here is a capillary, but this capillary is a glomerular capillary. It's still a capillary. Now, even though we haven't drawn them, there are fenestration slits along this capillary. There are poke holes along this capillary. Now, one difference is at this end, there is an afferent arteriole. At the other end, instead of a venule, there is another arteriole. There is an efferent arteriole. Now, up here, we have, instead of the interstitium, this is Bowman's capsule. Same thing down here at the other end. This is Bowman's capsule. Now, to help us envision that, let's go ahead and draw in Bowman's capsule. Afferent arteriole bringing the blood into the glomerulus, which is inside Bowman's capsule, and then the efferent arteriole carrying blood out of the glomerulus. The arrow is just showing us the direction of blood flow. So what we are looking at is going to be the pressures involved inside the glomerulus and Bowman's capsule. Now, these pressures will have very similar names. We have hydrostatic pressure. Now, this hydrostatic pressure is of the capillary, but remember this is a special capillary. So this is the hydrostatic pressure of the glomerular capillary. So we will just call it HPG, or hydrostatic pressure of the glomerulus. Now the afferent arteriole and the efferent arteriole, since they are arterioles, remember they have that ring of smooth muscle. They can adjust their vasodiameter, and they do. They can adjust their vasodiameter, which means they can adjust that hydrostatic pressure. They will maintain the hydrostatic pressure of the glomerulus at approximately 55 mmHg. Hydrostatic pressure of the glomerulus is 55 mmHg. Now we also have all of those solutes, primarily proteins, and again, primarily albumin. Remember, albumin is the primary protein that contributes to osmotic pressure in the blood. There are other proteins and other solutes that contribute, but in general, we just talk about the osmotic pressure. And the osmotic pressure, OPG, or the osmotic pressure in the glomerulus, is going to have an inward pressure in the opposite direction. So just like we did before, we're going to call that a negative pressure. We're going to call all outward pressures positive, all inward pressures negative. It's going to make the math easier, just like we did before. So osmotic pressure, we're going to give it a negative, and it is 30 mmHg. Outward hydrostatic pressure, 55 mmHg. Inward osmotic pressure, negative 30 mmHg. Now, what we have so far is we have blood passing through the glomerulus. A lot of that liquid portion of the blood is spraying out and filling up Bowman's capsule. Not all of it, because we have this osmotic pressure holding some of that liquid in place. Because the osmotic pressure is holding on to it. Remember, osmotic pressure, solutes suck. They are opposing that hydrostatic pressure. But as some of that liquid sprays out, Bowman's capsule is filling up. So as Bowman's capsule fills up with liquid, it's going to develop a hydrostatic pressure of its own. The hydrostatic pressure of Bowman's capsule, HPBC, or sometimes just HPC, hydrostatic pressure of the capsule, or capsular hydrostatic pressure. Bowman's capsule fills up with liquid. That liquid is pressing against the walls of its container, trying to press its way back in. So it's also going to be pressing this direction, the same direction as the osmotic pressure of the glomerulus. So we will call it negative. And it's going to have a negative pressure of about 15 mmHg. Now, so we have an outward pressure of 55, an inward pressure of 30, an inward pressure of 15. Will there be an outward pressure? Will there be an osmotic pressure of Bowman's capsule? Will there be proteins out here to pull liquids out? No, there should not be. Why not? Because proteins are large. Proteins are negatively charged. Large things and negatively charged things do not get filtered. So, in a healthy individual, there should not be any osmotic pressure in Bowman's capsule. There should only be these three pressures. So what we end up with is 55 mmHg, negative 30 mmHg, and negative 15 mmHg. We add those together and we get 10 mmHg of outward pressure. We call that the net filtration pressure, or NFP, net filtration pressure. Now, one other really big difference is when we did this for a standard capillary, we did this at the arteriolar end and the venous end. Well, there is not an arteriolar end and a venous end of a glomerulus. Here, it's arterial at each end. They both adjust their diameter, and the result is these pressures are constant the entire length of the glomerulus. So we do not have a difference at one end and the other. These pressures are the same throughout. So we do not have to calculate more than one for this glomerulus. We just calculate what we have right here. So here is how your book depicts that. Again, your book does not calculate positive and negative. It does this way. I think doing positive and negative is a little bit easier. You are welcome to do it whichever way you want. But up here, the result is the same. Here's our net filtration pressure, NFP, of 10 millimeters of mercury. Now, this is outward. That's exactly what you want. This means that we are pushing liquid out of the blood so that it can be filtered. We are filling up Bowman's capsule. As Bowman's capsule fills up, that liquid exits Bowman's capsule into the nephron, into the proximal convoluted tubule. We'll see what happens to that in our next lecture. We do not want a negative net filtration pressure. That would mean that liquid is going into the blood, and that would be fatal. That would be very, very bad. We want a positive net filtration pressure. Now, average net filtration pressure is 10 mmHg. It can go up or down, and it does go up and down based on the state of the body. Now, net filtration pressure is proportional to glomerular filtration rate, GFR. The glomerular filtration rate is how much liquid is moving out of the glomerulus into Bowman's capsule per minute. And that is about 120 to 125 milliliters per minute. Now remember, we have a lot of glomerulus. We have a lot of them. We have about 1 million nephrons per kidney. So this is not one nephron. This is not one glomerulus. This is all of the glomerulus. So we have about 120 to 125 milliliters per minute of liquid being filtered. Now, to tie this together, glomerular filtration rate is directly proportional to net filtration pressure. What that means is if net filtration pressure goes up, if it's above 10, well, glomerular filtration rate goes up. If net filtration pressure goes down, then glomerular filtration rate goes down. Let's take this back. If the afferent arteriole dilates and we let more blood in to the glomerulus, hydrostatic pressure goes up. It'll go from 55 to 60 or 65 or 70. So net filtration pressure will go up. So glomerular filtration rate will go up. If we constrict the afferent arteriole, hydrostatic pressure will go down. So net filtration pressure will go down, and glomerular filtration rate will go down. Now down here, the efferent arteriole, if we constrict the efferent arteriole, well, blood's going to get out of the glomerulus more slowly. It creates a backup. So hydrostatic pressure will go up. Net filtration pressure will go up. Glomerular filtration rate will go up. If the efferent arteriole dilates, now blood will get out of the glomerulus more quickly. Hydrostatic pressure will go down, net filtration pressure will go down, and glomerular filtration rate will go down. So NFP and GFR are directly proportional to each other. So hydrostatic pressure in the glomerulus is independent of hydrostatic pressure in the rest of the body, which is blood pressure. Hydrostatic pressure, or systemic hydrostatic pressure, is blood pressure. And hydrostatic pressure in the glomerulus is not related to blood pressure. They are independent of each other. That's extremely important. We do not want hydrostatic pressure in the glomerulus to be related to hydrostatic pressure or blood pressure. Because if net filtration pressure gets too high, we run the risk of damaging the kidney. We could tear those filtration slits. If net filtration pressure gets too low, well, now we're not filtering the blood, which is how we cleanse the blood. We need net filtration pressure to stay relatively constant so that we can cleanse the blood while keeping the kidneys healthy. So we have different ways to keep the pressure relatively constant. One way is something called the myogenic mechanism. Myo means muscle. Genic means generated. So muscle generated. The muscles generate that pressure. Smooth muscle reflexes. So we're going to draw this out a little bit so that we can understand it. So what we'll see is, starting with the drawing that we had just a moment ago, here is the glomerulus, but we're saying what happens if there is high blood pressure elsewhere in the body. But we need to make sure that we keep the hydrostatic pressure in the glomerulus the same. Well, think about what we've said about smooth muscle, or just any muscle in general. Remember what happens if you apply a little bit of stretch to muscle. What does muscle do if you apply stretch to it? Well, let's build on that. If there is high blood pressure elsewhere in the body, well, what's that blood going to do? It's going to come into the afferent arteriole and it's going to press against the walls of that arteriole. And if we didn't do anything about it, it would press harder against the walls of the glomerulus and it would increase that hydrostatic pressure. We don't want that to happen. So think back every time we've said, as far back as Bio 137 and earlier in this semester, when we said anytime that there is pressure applied or stretch applied to muscle, what does muscle do when there is stretch applied to it? It constricts. So when hydrostatic pressure or blood pressure rises, it's going to press against the walls of the afferent arteriole, and the afferent arteriole is going to vasoconstrict, which means that it's going to let less blood into the glomerulus. That blood is going to be under higher pressure because the blood pressure in the body is up. So less blood under higher pressure, it's going to balance out. So that hydrostatic pressure in the glomerulus will stay about the same. It's going to be under higher pressure, but less of it, so it should stay about 10 mmHg overall net filtration pressure. But what if there is less? What if blood pressure is lower? Well, the afferent arteriole instead would dilate to let more blood into Bowman's capsule. That more blood in under lower pressure would still balance out and keep that overall NFP about 10 mmHg. So that's that myogenic mechanism. The afferent arteriole constricts or dilates based on the amount of stretch. So here is, again, remember when I said this is the way that I draw a nephron, this is the way your book draws a nephron, this is the way a nephron is always going to be presented just because it's really easy to draw. But this is more how a nephron actually looks, and I said that there's actually a reason why it's actually important to draw it the way that we draw it, because we have to point to things, but there's importance to how it actually appears. Well, this area that we've got circled right here, there's something really, really important that happens here. Now, I know we haven't really talked much about the nephron yet. You have read about the parts of the nephron in that self-study part. There's something called the distal convoluted tubule and the loop of Henle. And right where the loop of Henle and the distal convoluted tubule, right where they meet, they bump up against the efferent arteriole. And that happens right here. And there's something really important about that area. So right here is that part where the loop of Henle becomes the distal convoluted tubule. And right here is the afferent arteriole, right here is the efferent arteriole, and there's some special cells right here called the macula densa. And the macula densa and some of the cells of the afferent arteriole, some of the cells of the efferent arteriole, they all work together, forming something called the Juxtaglomerular apparatus. Usually it's abbreviated the JGA, but the JGA, or the Juxtaglomerular apparatus, is also going to control a little bit of this filtration process. Now, this part of it is going to make a lot more sense after we get through all of these lectures. I'm going to talk about it right now, but it's not going to make sense. So don't worry when this doesn't make sense. What we're going to do is talk about it and then refer to it again at the end of these lectures. It won't make sense now. Don't worry about it. It will make sense after we get through the lectures. So, another way that we can regulate filtration is something called the Tubuloglomerular feedback mechanism. And it uses that macula densa. The macula densa has chemoreceptor cells, and those chemoreceptors detect concentration of sodium chloride, salt. Now, as that filtrate moves through the nephron, specifically as it moves through the loop of Henle, this big U-shaped area right here, Well, as it moves through there, water and salt are being reclaimed by the body. Now, the quicker it moves through, the faster, the quicker it moves through, the less chance sodium chloride can be reabsorbed. So let's think about what that means. If we are filtering very, very quickly, that's pushing everything through very, very quickly. So we don't have as much chance to reabsorb everything that we might need to. So that affects the concentration by the time we get to the end of the loop of Henle. Now those JG cells, the juxtaglomerular apparatus, samples that filtrate. And based on the concentration, it can say, hey, we are filtering too quickly, or we are not filtering quickly enough because of the concentration of sodium chloride. So we can either filter more or less quickly. And what it will do is it will dilate or constrict the afferent arteriole so that it adjusts the net filtration pressure, causing us to dilate and causing us to filter faster or slower based on what it judged of that concentration. Now again, I know that does not make sense right now. Once we talk about the loop of Henle and how the loop of Henle works to reabsorb water and salt, come back to this and it will make more sense. So, that is autoregulation, the tubuloglomerular feedback mechanism and the myogenic mechanism. But autoregulation works between 80 and 180 mmHg. Outside of that, it is overridden by extrinsic controls. Extrinsic controls just means things outside of the kidneys. The kidneys control within that range. If MAP is outside of that range, then that means things are dire and we need extra help. We need help from outside the kidneys. And SNS will take over, for example, in states of hypovolemic shock. If we have lost a lot of blood. Well, if we've lost a lot of blood, we do not need to be thinking about removing more liquid from the blood to make pee. So the sympathetic nervous system will constrict the afferent arterioles, basically cutting the kidneys off from making urine. That decreases net filtration pressure and glomerular filtration rate. It almost cuts them completely off. Why? Because we don't want to urinate, lowering blood volume even more. Also, the renin-angiotensin mechanism. Remember the renin-angiotensin mechanism? Go back and look at your renin-angiotensin mechanism again. We drew that out a couple of times now. But the renin-angiotensin mechanism, now when we draw it, we don't just say renin is released from the kidneys. We're going to be a little bit more specific. If I ask you to draw the renin-angiotensin mechanism, now the renin is released from special cells called JG cells or juxtaglomerular cells. And we're going to be more specific. Before, we just said when blood pressure drops, but we're going to be more specific. Now we're going to say sympathetic nervous system stimulation causes renin to be released from the JG cells. Or when macula densa cells are stimulated, renin is released from the JG cells. Or when there is a decrease in stretch because of a drop in systemic blood pressure detected by the afferent arteriole, renin is released from the JG cells. So now that renin-angiotensin mechanism has come back. But it is coming back with much more detail. So here, this now sums up everything that happens when there is a drop in systemic blood pressure. There is a lot that's going on. This sums up everything that we have talked about so far in this course when there is a drop in systemic blood pressure. This is from our textbook, so it talks about things that we've talked about in this chapter, but it talks about things that we have talked about previously. There's a lot going on. Make sure to review this because it does pull together things from much earlier in this chapter, or much earlier in this course. All right, so that brings us to a pause point now. Next thing we will be talking about is, now that we have filtered and we've produced filtrate, that filtrate is going to move through the nephron. We're going to look at reabsorption and secretion. We're going to look at how do we reclaim stuff from that filtrate to put it back into the blood. We're going to look at some of the stuff that was not filtered. How do we put it into the filtrate? Then we're going to have a lecture over what happens to stuff as it moves through the loop of Henle. How do we maximize water reabsorption? All right, so make sure to go through this lecture a time or two. Filtration is a really big process. Make sure to understand it. Reach out if you have questions. All right, take care, and I'll talk to you next time.